Okay, first I want to thank the organizers for giving me the opportunity to present our project today, and of course the CRAFT COVID committee for funding our research. So this project is focused on improving and streamlining the testing pipeline, and it's a collaboration of Phil Falkner and Rafael Ramiro de Assis at the Vaccine Research and Development Center at the School of Medicine, and me at the Laboratory for Fluorescence Dynamics of the School of Engineering, and also Wayne Zhao, who will be the next speaker at Pharmaceutical Sciences. So as you know, there are basically two ways to test. One is direct detection of the virus or virus RNA, typically done by PCR testing, and then there's antibody testing to study the immune response, and both methods have their specific strengths and weaknesses. Of course, if you're directly measuring the virus, this is the earliest time you can detect it. However, if a patient has overcome the infection, and we know there are many asymptomatic carriers, by PCR testing, we will not know if this individual was infected or not. And this is what antibody testing can deliver, because these antibodies build up and will remain in the sera for months and years. And this will be very important in the next couple of months and also years, as we want to study what is the percentage of the population that has had the disease, to plan further containment and mitigation measures. And also, as we want to, of course, develop vaccines, we also need to study the antibody response, and we need tests for that. As you know, some of the antibody tests that are now available have received some critiques that they are not very accurate. And for example, those simple lateral flow assays, they often only test one antigen at a time, and you also only do one repeat at a time. So this, there's a chance that you have false negative, false positive. So an alternative to these approaches are microarrays, as Greg already mentioned in his very nice talk. So here, the idea is that we can deposit proteins, in this case antigens, in micro-sized dots, about maybe 100 microns in diameter, on the surface of slides or well plates. And this way we can have tens or hundreds of antigens in the same array, and even replicates in the same array, and probe many at the same time in many patients at the same time. So these can be printed. For example, in Phil's lab, there are 3 of those printers. You can see a lot of slides can be printed in a relatively short amount of time. Then the probing can be done in a clinical lab, where the blood samples are added, and then the secondary fluorescently labeled antibodies. And then we have an imaging step where these antigen arrays are read out, and then of course they can be analyzed and used for different studies. So in Phil's lab, they have recently constructed a coronavirus antigen array that includes the SARS-CoV-2. They recently submitted 2 manuscripts about this coronavirus microarray, which has a very high sensitivity and specificity. So here you can see all the antigens that are probed in this array, and here most importantly on the left, 7 antigens to SARS-CoV-2. As you can see, the red ones are the positive samples, and the antigens have different responses, some stronger, some weaker. But for all of them, you can pretty well distinguish them from the negative. And the nice thing about this panel is that it also includes many other respiratory viruses, so the regular coronavirus, the SARS, the MERS, common cold, influenza, and many more. So you really get some comprehensive information that will also be very important for clinical studies. So to summarize, we can test hundreds of antigens simultaneously in a single test, high specificity sensitivity. And those tests, importantly, contain information that can be used to design mitigation measures and also for vaccine development. So in this whole pipeline that you've seen in the project, we focused on the imaging part. And just to show you some instruments that are used right now, there are these laser scanners. Those are pretty dated designs going back to the 90s and early 2000s. They take about 10 minutes to image a small 75 by 25 millimeter slide. Very heavy and very expensive. And they are not typically present in clinical labs. Then technology has improved. So there are now these camera-based systems, which are faster, way less, also less expensive. Still, the typical clinical lab doesn't have these kinds of devices to be able to make use of the microarrays. So for this, we wanted to develop a new imaging and data analysis platform based on a very compact design, also using commonly available components and 3D printing, so we could quickly mass produce it and distribute it and establish this testing method at clinical labs, and then pair it with a platform where you can instantly upload the data to a server where it will be automatically analyzed. So to have a comprehensive, large-scale analysis platform. You can really see the scale of these images now. It's amazing. You can shrink them down to basically the size of a smartphone. Yeah, so we already did some testing. Here you can see example images taken with this miniaturized device, and we compared it with a commercial imaging system and get a very good overlap of the data. And here on the slide, you can see patient samples that we used to probe the microarrays, and again, they were both imaged with the commercial and our new imaging solution. Again, very good overlap between the data, and down here, circled with the orange circle, you can see the relevant SARS-CoV-2 samples, the negative group, towards the positive group, which can be clearly resolved with both systems. So in conclusion, this imaging system can provide very accurate results using this very compact, modular, and easy to use designs. During the project, we will now also go towards developing this cloud-based analysis I was mentioning, and we are going to integrate this imaging platform after further optimization with large-scale serological testing platforms, so we can really scale up the testing. And I should mention that for this, we also are partnering with a company in Irvine called ResearchDX, because as you can imagine, in an university labs, it's very difficult to do like thousands, 10,000s, or even 100,000s of samples, which we will be getting. So Phil's lab is receiving, I think, 5,000 samples next week here from the local hospital. I also would like to mention that we have already prepared a manuscript about this. I'm collecting the edits from co-authors, and we hopefully will be able to submit even today. And we are also preparing an R1 application to NIAID, where we will focus on this large-scale serological testing based on microarrays. Again, I'd like to thank, of course, you for listening, and of course the UCI Craft COVID initiative for providing funding for this research. With that, I'll take any questions.
Okay, thank you very much. I could start out with one, actually. Could you just, what is the cost of the smaller array scan camera supposed to be, roughly? Do you know?
So the components we're using are about $100, $200 maybe.
So I noticed you're not mentioning the price, but that's okay. So, and then on the larger scale system, is that a scanner or is that just a built up CCD camera? What?
So these are what we're using is just cameras like you're using in your phone, essentially.
But for the larger scale, it's just
for the larger scale, we'll still be use cameras, but instead of using those 2x8 slides, the microarray slides that you can see here, we will scale up to 96 well plates. And to image those, we need to take a few images. So in this case, we'll still use a camera, but we will move the assembly in 2 or 3 steps to image the whole plate.
Yeah. Okay, thanks. Thank you. Do we have questions from the audience?
Yeah, just a comment that, you know, that as the coronavirus epidemic still rages in India and is really taking Africa by the throat right now, this would be very attractive to develop overseas as well. Niklas, I'm guessing from your name that you're Scandinavian, maybe Swedish. Is that right?
Originally, I'm from Germany, but my mother was born.
Okay. Well, Europe still has a good relationship with the rest of the world, so it might be a good idea to look for a company to partner with over there, you know, in terms of putting this on the market. Because often it's a cost of equipment that really handicaps people in the third world.
Yeah, actually, I have a friend and collaborator in South America, and he would be very interested in this too.
Yeah, the Africans are easier to deal with.
It's
Good luck.
Mike has always been very international in his perspective. Sid, did I?
Yeah, yes. Just following up on Mike's line of questioning here. How much training does it take for somebody to learn how to use this camera? Is it the kind of thing where you can train somebody and they can go into, let's say, a nursing home or a prison or some place that needs testing right now in the US and gather the information?
Yeah, so using the camera is, I would say that's more or less the easiest step. What still needs to be done is probing the array. So you need to add your blood sample and you also need to add your secondary antibodies and probably do a washing step. So this is the more labor-intensive part.
But with respect to that, I think something not everyone appreciates is yours is one of the few available, I won't say FDA approved, but available test that's a pinprick. Is that right? As opposed to a blood draw.
Yeah, so
that's a big deal.
The sample you need is only a microliter, so you can just do a finger prick. Yeah.
More questions? Okay, thank you very much.